You sit the same, everybody's fine where you're sitting? So plenty of other chairs if you need to move to a chair so you can see better. Everybody's fine. Okay, great. So I'm just going to go off of this review sheet. And again, if you don't have the review sheet, feel free to come up and get a review sheet. I have some extras here. The first activity I want to talk about is the scientific method. And if you have your lab manual, a great reference page for the scientific method in our laboratory manual, I'm looking, is page 3. And so number 1 is outline the steps of the scientific method. So what is the first step of the scientific method? Go ahead. Observation. This is step number 1. Can you guys see this okay?
Yeah.
Okay. It says new observations are made and previous data are studied. Number 2, step number 2. I'm sorry?
Hypothesis.
Hypothesis, excellent. And in our book it says input from various sources used to formulate a testable statement. Remember a hypothesis, many of you have learned it's an educated guess. That works okay. Or a hypothesis could also be defined as a tentative explanation. A tentative explanation, hypothesis. Okay, step number 3. Experiment. Also known as testing. Step number 4. Conclusion. Now remember, your conclusion can support your hypothesis or not support your hypothesis. If the conclusion does not support the hypothesis, typically we go back to the hypothesis and maybe rewrite the hypothesis, or we go back into the experiment and see if maybe we made some errors in the experiment before we just disregard the hypothesis altogether. But a conclusion can either support your hypothesis or not support your hypothesis. Okay, I have number 2. If given an experimental variable, be able to determine experimental group, control group, experimental variable, and data. So let's just use an example that we did. The one that we did that I want to reference is the experiment we talked about in lab that had shrimp involved. You guys remember that one?
Yeah.
And we made the observation that shrimp color varies. And so I came up with the hypothesis that shrimp color is due to the algae color, the color from the algae that shrimp eat. And then if you recall, we decided as a group to set up 4 aquariums, put shrimp in all the aquariums, and then put brown algae in one aquarium, green algae in a second one, red algae in a third, and shrimp chow in the last one. Does everybody remember that okay? All right. So based on that, I think we should be able to figure out these 4 things up here. Based on the shrimp experiment, what were the experimental groups? Which tanks? The first 3, the ones that ate the brown algae, the green algae, and the red algae. Is everybody okay with that? Those are the experimental groups. Okay, what were the control group? Or what was the control group? The tank that was eating the shrimp chow. How come that was the control group?
Because we knew what outcome we were going.
Excellent, Diamond. She said on a control group, you're supposed to know the outcome before the experiment ever starts. So the control, you always know the outcome. Now, let's just diverge a little bit here on the control. Every good experiment has a control in it for 2 reasons. 2 important reasons that every experiment has a control. Can you come up with a reason why you have to have a control? To compare, excellent. To compare and to validate. Excellent job, Erica. Excellent job, Nyla. To compare and to validate. I will ask you that. What are the 2 reasons that an experiment has a control? To compare, to validate. It's very good. And remember, Diamond said that your control group, you should know what's going to happen. What if what you think is supposed to happen in your control group doesn't happen? Then what's going on with that experiment? Is it a valid experiment? No. So that's what I mean by the validation. If the control group doesn't come out like you're expecting it to come out, then your experiment's not valid. Not valid. Everybody okay with that? Control group? Okay, great. Thinking about our shrimp experiment, what was our experimental variable?
The feed.
The food, the diet, the food. Remember, one aquarium got brown algae, one got green algae, one got red algae, one got shrimp chow. That was the only thing that was different between the 4 aquaria. What is different between the groups is the experimental variable. What differs between the groups? Experimental variable. Okay, then lastly, during the experiment, we collected data. The data that we collected were things like mortality of the shrimp, the color of the shrimp, the pH of the water, the temperature of the water. So any information you collect during the experiment is called data. But okay on that word data? Okay, is there anything up there that you have a question about? Any of those 4 bullets that you have a question about?
It's kind of like you're referring to the shrimp example.
Yes, so referring to the shrimp example, the experimental groups were the red algae group, the green algae group, and the brown algae group. And the control group was the shrimp chow group, so it was all referencing that shrimp experiment. Yes, ma'am? Okay, so what Jordan's saying is, Ms. Steinke, the shrimp questions ought to be on the test because we already went over that twice. And so she's right. I'm going to come up with another experiment that I describe, and then you have to figure out who's the experimental groups, who's the control group, why do we have to have a control group? Give me 2 reasons why. What's the variable? What's the data? Okay, so be able to analyze an experiment and tell me these things. It's a good question, Jordan. Anybody else? Okay, super. Okay, be able to form a hypothesis. Now, one day, I don't know if you remember this, one day you came into lab and we had earthworms. They were alive. Do you remember that day?
Yeah.
And we were doing scientific method using earthworms, and we practiced writing hypotheses that day. So somewhere in your folder, your notebook, you kept that experiment. Just go back and read how we wrote hypotheses. And then I also remember at the end of that activity, we practiced looking at sentences and being able to pick out which would be a hypothesis. So make sure you review that lab that we did. It was called Scientific Method, and it was using the earthworm. Okay, if I give you some data, you should be able to reach a conclusion. I have no worries about this one because every day you and I do experiments. We talk about them, and then we reach conclusions. Usually the experiments are in test tubes, and we look at our test tubes. We talk about what we got in our test tubes, and then we reach a conclusion based on that. So I'm not worried about this one at all. You're quite accustomed to doing that. Okay, so does anybody have a question about Exercise 1 Scientific Method? Okay, you can see I'm going to kind of go fast because we have a lot to talk about. So I'm going to move on to Exercise 2, Metric Measurement. And if you're following along in your lab manual, metric measurement starts on page 9 and then moves on over to page 10, etc. So we're going to spend a little bit of time here with the metric system. Number 1 was know the meaning of the metric prefixes. So what does that really mean? What does Ms. Steinke want out of me? Well, this is what she wants right here. I want you to be able to fill in these blanks. Take a look up here if you can't see the screen. You should be able to fill in these blanks. Fill in the blanks. Okay, so let's talk about this first blank. How many centimeters in 1 meter? 100. Who told me that? Very good, Adam. 100. But okay on what Adam said. How many millimeters are in 1 centimeter? 10. Who told me that? Very good, Catherine. 10. So let's see if you can logically figure this out. Based on the first 2 statements, how many millimeters are in 1 meter? 1,000. That's good, Diamond. 10 times 100, 1,000. Everybody okay on that? Okay, now, buddy, listen. You one day took out a microscope, put a ruler on the microscope, and we measured the field of view of the microscope using the ruler. And you may remember that on the lowest power, the field of view was 5 millimeters. And then we went up to the next power, and it went down to 2 millimeters. And we went all the way to high power, and it was so small that we couldn't even see a millimeter anymore. So we mathematically figured out that the field of view was half of a millimeter. And so I think you'll agree with me when I make this statement. Millimeters are not very useful under the microscope because they're too big. But you follow with me. Millimeters would not be very useful to do length, measure stuff. Under the microscope, they're just too big. So check this out. And I want you to write this down. I made an error right there. I had to cross it out. So what happened was, with the microscope, it didn't take us very long to figure out millimeters are not very useful under the microscope. So we had to come up with another unit that was more useful. And it's called the micrometer. Now, this is the symbol for the micrometer. It's kind of a funny looking U. Why didn't they just use MM for micrometer? It's already used. Thank you, Jordan. It was already used. You couldn't use it because it's already used. So micrometer is going to be that funny symbol and then a little M. Because this funny little symbol that looks like a U means micro. If you don't like how I'm saying it, micrometer, you could say it this way, micrometer. Maybe you like it that way, micrometer. Okay, so here's what they did, guys, watch. They took a millimeter and they divided it into 1,000 equal units, and that's what they call a micrometer. It takes 1,000 micrometers to make 1 millimeter. And that was working out great until we invented the electron microscope. And then with the electron microscope, the micrometer was too big. It's just too big. So they had to come up with an even smaller unit. I know you've heard of this NM before. What's the N stand for? Nano. So what they did was they took 1 micrometer and they divided it into 1,000 equal units. And they call those nanometers or nanometers, whichever you prefer. So the question is, how many nanometers are in 1 millimeter?
10,000.
Nope. Okay, well, let's see. How did we get 1,000 on this screen? Everybody remember this previous screen? 100 times 10. So how are we going to get the answer? 1,000 times 1,000. What is 1,000 times 1,000?
No.
It is. Thank you, Saul. It's 1,000,000. Oops. Ooh-wee, that's little. It takes 1,000,000 nanometers to make 1 millimeter. That's little. So let me just tell you when you're going to see these things. You're going to see micrometers, this one used, whenever you're just measuring stuff using a compound light microscope, like the size of a nucleus or the size of a chloroplast. You're going to start seeing NM when you're using an electron microscope. Really little bitty things, like ribosomes, they'll measure in nanometers. Or molecules, they measure molecules in nanometers. Viruses are really small. You guys have heard of viruses like the Ebola virus. That's a virus. Everybody knows that's not a cell, right? You guys know viruses are not cells, right? Shake your head. Viruses are not cells. Okay, and so the Ebola virus, if we were looking at it underneath the electron microscope, we'd measure it with nanometers. So really small things, you use nanometers. Nanometers. Because nano means really small. Micro means small. Nano means really small. But okay with that? Okay, good. All right, so based on what we just did, this is question number 2. We're going to do these conversions. So I have 23 centimeters. How many millimeters would that be?
230.
Excellent. Who told me that? Nyla? Good job. 230. She moved the decimal place one time to the right. Okay, 368 millimeters would be how many micrometers?
368,000.
Boy, that Nyla, she's on fire on these conversions. Okay, 6 micrometers would be how many nanometers?
6,000.
6,000. Okay, I just want to comment on how Nyla apparently does this very well. If you're not seeing where she's getting this stuff at, it's from the previous screen and the one before that. What she was thinking is, well, 1 centimeter is equal to 10 millimeters. So 23 centimeters is equal to 230. She moved the decimal place one time. Don't you just move the decimal place one time to the right to go from 1 to 10? So she moved the decimal place 1 to the right to go from 23 to 230. Okay, and then if you look at the previous page, she saw that 1 millimeter is equal to 1,000 micrometers. So to make 1 become 1,000, you have to move the decimal place 3 times to the right. So that's what Nyla did. She moved the decimal place 3 times to the right. Same thing here. Nyla saw that 1 micrometer is equal to 1,000 nanometers from the previous page. So she just moved the decimal place 3 times. So these top little equalities kind of tell you how come Nyla made these decisions to move her decimal place, in case you weren't sure how she was doing it, because she was doing it very well. Very good, yeah. Anybody have a question about the conversions? Nobody? Okay. All right, in your drawer, open up your drawer. There should be a little plastic ruler in there. Might be clear or it might be white. You guys have a ruler? Okay. Is there anybody who needs a ruler? I have extras. Anybody else need a ruler? Just Catherine? Okay, so on the ruler, make sure you're looking at the metric side. The metric side. How many centimeters are on this ruler? 15. It's Michaela. Yay. Everybody agree with Michaela? Okay, how many millimeters are on this ruler? 150. Michaela, how'd you get that? Because we know that 1 centimeter is equal to 10 millimeters. Or I can just see it right there on the ruler, that each centimeter is divided into 10 equal units. And the little units are millimeters. Everybody know those little black lines are millimeters? Yes? Okay. Everybody feeling comfortable with the ruler? Great. Question 4, what are the freezing and boiling points of water in Celsius? I'm only interested in Celsius. What's the freezing point of water in Celsius? 0. And boiling? 100. Is that you, Alex? Yes. Good job. Do they have a question about those? You have to know the freezing and boiling points of water in Celsius. Okay, this was from our very first day together. We worked as a team, and you're going to have to do some of the skills that your team did that day. Let me tell you what you're going to have to do. You're going to have to be able to measure a bone, and you have to measure it in length using a metric ruler. Measure the length of the bone. Shake your head. I can do that. You have to measure the mass of the bone using the triple beam balance. You have to do volume, not of a bone though. And you have to do volume in two different ways. Let me talk about the volume. The day that we did these activities, there were two things on your table that you had to do volume on. Remember, volume means how much space you take up. They were a seashell and a wooden cube. I don't know if you remember that. It was a seashell and a wooden cube. Does anybody remember how you figured out the volume of the seashell? It fit into a graduated cylinder. So everybody shake your head. Graduated cylinder. Now, question. Do we put water in the graduated cylinder?
Yes.
Before or after the shell? Before. And do we have to know how much water is in there before the shell?
Yes.
Yes. Okay, so I'm just making this up. Let's say I put 25 milliliters of water in the graduate cylinder, and then I drop the shell in, and it goes to 29 milliliters. What's the volume of the shell?
What was it before?
25.
Oh,
And it went to 29.
4.
It's 4. So how'd you figure that out?
It's 25 minus
29 minus 25. So it's how much did it go up? It's the difference, right? But okay, I'm doing volume with the graduated cylinder. Okay, on the boggle cube, I purposely made it where the boggle cube wouldn't fit into the graduated cylinder. You may remember that. So how did you get the volume of the boggle cube? Length times width times height. And that's why you need a calculator during the test on Thursday. Length times width times height. So just to recap, on volume, you have to be able to do volume two ways. One using the graduated cylinder, and one using length times width times height. Okay, next question. What is a meniscus? And I put a little drawing up there. Isn't it cute? Okay, so Denise, you're saying there's lines on the graduated cylinder, and then what else? What else do you want to say about that? Okay, so I'm going to just color. Denise, what I have coloring pink right there, what's that supposed to represent? Liquid. And everybody notice the liquid isn't going straight across. The liquid is doing this at the top. And so where it dips down, we call that the meniscus. And you guys remember why. The liquid is usually water. And water is adhesive to the glass. So it's sticking to the sides of the glass, and that's what makes it go up on the sides. And so what's the proper way to read the meniscus of a graduated cylinder? Do you read the top? Do you average it? Do you do the bottom? What do you do? The bottom. You got to read the bottom. Okay, here's what I do not want to see during the test. Do not pick up graduated cylinders. What do you have to do? Lower yourself. Very good. You got to get down eye level like Deanna just said. You leave it on the table because that way it's level. And then you get down eye level. Now, the lab test is multiple choice. So I'm just making this up. Let's say that I had a graduated cylinder, had 66 milliliters of water in it, and I said, how much water is in this graduated cylinder? Well, 65 is a choice, and 66 is a choice, and 67 is a choice. So all the ballpark numbers are choices. So you really have to take your time and read it properly. And if you're trying to read it up here, you're going to mess up. Okay, so make sure you get down eye level and count the lines and make sure that you're right. So take your time on that. Okay? But it's okay on the meniscus question. Super. All right, moving on. Exercise 2 is microscopy. Let's start on page 14. Okay, on page 14, there were some pictures that I spent a little bit of time talking to you about. On page 14, letter A, B, and C. So I just want to comment on those. With this question number 1, what microscope would you use to view different things? And so if you just wanted to see blood, like letter A, you would just use a compound light microscope. And you maybe already wrote that down for letter A in your book. That's a compound light microscope. Just a regular microscope like what we have in class. Compound light microscope. Got to go, Jordan?
Yes, ma'am.
It's okay. And this was pretty good magnification right here, because I remember last week I showed you blood. I don't remember who was letting me get blood out of their test tubes last week. It was right here. Okay, and you remember those cells are real small, so this is pretty good magnification for letter A. All right, letter B, that's still a blood cell, but man, that's way bigger than before, and look how I'm seeing all those internal details. That's an electron microscope right there. It's called a transmission electron microscope. Look at all the details there. I can even see the mitochondria, the nucleolus. Wow, that's really good. Letter C is an electron microscope also, but I'm not seeing inside the cell. What am I seeing on letter C? The outside of the cell. So on number 1, make sure that you are looking on page 14 and seeing how you can see different things with different kinds of scopes. Which reminds me of question 2, how are light microscopes and electron microscopes different? Well, obviously the light and the electron make them different. Light microscopes use light as a source of energy. Electron microscopes use electrons as a source of energy. Is everybody okay? Well, just that obvious thing? Okay, what about on magnification? Which one's higher in magnification, light or electron? Well, look at the pictures on page 14. A is light, B and C are electron. Which one has higher magnification? Makes things bigger. Which ones make things bigger? Electron. So we have much higher magnifications. If you're curious as to where can I go for information about this, page 15, table 2.2. It's page 15, table 2.2. Be a great place to look at the differences between compound light microscopes and electron microscopes. Okay, a dissecting scope, page 16. I like dissecting scopes. They're also called stereoscopes. I wrote that in. We use these twice. So far, we've used them twice. One day, we just looked at all kinds of stuff. Some of us looked at a moth, some of us looked at a butterfly, some of us looked at plants, some of us looked at a feather. Somebody in here looked at a starfish under the dissecting microscope. And then our day that we spent together with the earthworm, we should have all looked at the earthworm with the dissecting microscope. Now, in your opinion, does the dissecting microscope show internal details or surface features? Surface features, very good. Surface features. So it's like dissecting microscopes are really nice magnifying glasses. Just really nice. Deluxe. So if all you want to do is see surface features, this is the microscope for you. Okay, star number 4, because this one's going to have a lot of questions. The parts and the functions of the parts of the compound light microscope, page 18 and page 19. This is page 18 and page 19. And you want to study, study, study. This is real important. Okay, on number 4, you see it on page 18 and 19. I think we filled it out together there. Is okay on that? Super. Number 5, know how to use the compound light microscope on low and high powers. Well, I put a little drawing up there. Guys, this is supposed to represent the circle of white light that you see when you look through a microscope. Don't you agree when you look through a microscope you see a circle of white light? And then we call that circle of white light field of view, FOV, field of view. Now, here's what I think. I think you and I have figured out that when we first start with the microscope, we always go to 4x, the short one. Haven't we figured that out? And then we focus on what we want. We put it in the middle of our field of view, and then we go to 10x. And we find what we want, we put it in the middle, focus it, adjust the light a little bit, but we always put it in the middle, and then we go to high power. And then when we get to high power, we have to kind of think about it, because I remember this, the high power objective is so close to the slide that I only get to use the fine adjustment. I'm only allowed to use the fine adjustment when I'm on high power. Do you guys remember that? Okay, so I want to know, logically, why is it that you and I always start on 4x? Why do we do that? Tell me again, Denise. You can see more. So using this FOV idea, field of view, it's because 4x has the biggest field of view, because Denise just said we can see more because we have a bigger field of view. So it just makes sense. Okay, you can see more so you can find what you want. Okay, so let me just repeat. Know how to use the compound light microscope. Well, start on 4x, find what you want, put it in the middle, make sure it's focused, and then you move to 10, find what you want, put it in the middle, make sure it's focused, then you go to 40, find what you want, put it in the middle, and when you focus, when you're on 40, you're only allowed to use what? The fine adjustment. So I'll ask you that. What adjustments can you use on high power? Only the fine. What adjustments can you use on the 4x scanning? Both. What adjustments can you use on the 10x low power? Both. What adjustments can you use on high power? Only the fine. Is everybody okay with that idea? Okay, total magnifications on page 21. There's a table there. Could you glance at that table? We have completed it, of course. But can you find the general summary of an equation that you could tell your third grade friend on how to calculate total magnification? What do you do? Okay, so say it louder, Deanna.
10 times whatever the number is on the phone.
Okay, so why 10? Okay, so Deanna said, look at the ocular. Now, when you're looking at the ocular, how do you know what the magnification of the ocular is? Because darn it, there's a bunch of numbers on there. Which number is the magnification? It's got the 10, and what else does it have? X. Looking for the number that has the X. And Deanna said for our microscopes, it's 10. And then she said, then look at what the objective is that's clicked into position. And again, look at that objective and find the number that has the X. And then you're going to multiply it. Now, if we were on scanning, you'll see this in your book there. If we were on scanning, you would have found a 4. So, take a look in your book. Did we go 10 times 4 for total magnification of 40? Is that true? So we must be multiplying. So the basic equation for total magnification is ocular times objective. Ocular times objective. It's such an easy question. Look at this one. I can't even believe I asked you. Which objective would give the largest field of view? 4. Also known as scanning. And guys, we actually proved that. I don't know if you took notes about this on page 21. We did the diameter on scanning, which I think came up to 5 millimeters. Then we did the diameter on low, which I think came up to 2 millimeters. And then we did the diameter on the high power, which I think came up to 0.5 millimeters. So we proved that this was true. Lower magnification, higher field of view. Lower magnification, higher field of view. Are they okay on that one? So easy. Okay, inversion is page 20. Let me look at page 20. Okay, so let me show you inversion. We use that to do inversion. What is that?
Backwards.
Yeah, that's the letter E. And that's what it looked like before we put it on the microscope. And then when we put it on the microscope made it bigger, right? Because that's what microscopes do. And what else did the microscope do to it? It flipped it and reversed it. And flipping and reversing is called inversion. Inversion. Microscopes, compound-like microscopes flip and invert, completely make the top, the bottom, the left, and the right because of the mirror. And again, inversion's on page 20. If you want to reference a page in your notes for number 6, page 20. Speaking of inversion, how does inversion relate to microscope usage? If you push a slide to the left, what way does the image go? To the right. If you push a slide down, the image goes up. So what's the general statement here? All movement is opposite under the microscope. So I'll write that down. Movement is opposite. Up goes down, left goes right. And again, it's because of the inversion. If I have a question about exercise 2 on microscopy, the part on microscopy. Nobody? Everybody's good? Okay. Exercise 3, the chemical composition of cells. Let's all go to page 40 in our book, page 40. We're going to find lots of answers there. One place. Page 40. Here we go. Question number 1. What is used to test for protein? I'm ready. Biuret. Very good. Biuret. What's the positive color using biuret for protein? Purple. Thank you. Or a pinkish purple. What is the negative color using biuret for protein? Huh? Blue. Thank you. Did I see where I'm getting this from? Page 40? Yes. So I'm thinking I have to know page 40. Next one. What is used to test for starch? Iodine. Good. Positive color.
Dark colors.
Yeah, it could be chocolate brown, could be navy blue, dark purple, black. So dark colors. What's the negative color?
Amber.
Amber, thank you. What is used to test for sugar? Benedict's and heat. We've got to heat that one. So I'll write that down. Okay, what's positive color?
Green, yellow, orange.
So if we got green, that means there's a little bit of sugar. If we got yellow, it's a moderate amount of sugar. And if we got orange, man, do we have some sugar. What's the negative color? Blue. Okay, Diamond better not be the only one who knows this. I'm getting all this off of page 40. Page 40. Is okay so far? Okay, great. How do you use brown paper to test for lipid? Well, I know we put something on the brown paper, put it in the microwave to evaporate the liquid. You guys remember the microwave part? And then we hold the paper up to the light. And what are we looking for?
Translucent.
Very good. I like the way you're using that word. Translucent means that light can come through it. So a translucent spot means yes. Yes, you do have lipid. What if you get no translucent spot? No, you do not have lipid. You can get a spot, but if no light's coming through it, then no for lipid. I'll just give you an example. Some of you on your unknown, you may remember you got starch. Some of you had starch. And so you did get a white spot on your brown paper, but when you held it up to the light, no light came through. Because starch will leave a spot, but not a translucent spot. Okay, on the brown paper test. Okay. All right, so let me give you some page references for our next questions. The building blocks of carbohydrates are back on page 28. What are the building blocks? Looking on page 28, what are the building blocks of carbohydrates?
Glucose.
Glucose is an example of what?
Molecules.
Monosaccharides. Very good. Monosaccharides. I'll take monosaccharides for an answer. Here's another possible answer. Simple sugar. They're the same thing. Monosaccharide equals simple sugar. I'll take either answer, monosaccharide or simple sugar. Those are the building blocks of carbohydrates. It's like this is the smallest thing that we could call a carbohydrate. Okay. What are the building blocks of lipids? Well, lipids are over a couple pages. Page 34. We'll look at page 34 for lipids. Page 34. What are the building blocks of lipids? Everybody take a look at page 34. Go ahead. Huh? I don't hear anything. You on page 34? How do you build a lipid? Go ahead.
What do you mean, like the fats and oils?
At the bottom of page 34, formation of a fat. What are the blocks you put together to make a lipid? Glycerol. Thank you, Kaylin. Glycerol. You found it now. And what else? 3 fatty acids. That's very good. But he's okay on that. Building blocks of that. Lipid, page 34. Let's see what's next. Building blocks of proteins. Everybody knows this. Sure. Very good, Deanna. Amino acids. If you need a page number for that, check out page 33. If you want a page number for the building blocks of proteins being amino acids, page 33. Okay. Okay, everybody knows this question. Monomer means 1 unit and polymer means many units. So I want to know, how do you take, we'll just do, if everybody's on page 33, let's just do amino acids. Amino acids would be the monomers, and dipeptides, polypeptides, and proteins would be examples of the polymers. So what I'm asking you is, what kind of chemical reaction are you doing if you're taking small monomers and putting them together to make them larger? Page 33. What kind of chemical reaction are you doing? Thank you. Who told me that? Very good. Lindsay said dehydration. Taking small things and putting them together to make larger things. It's called dehydration. Everybody look at the picture. Taking amino acids and linking them together. Dehydration. Everybody see the arrow with dehydration on it? You're okay with where Lindsay found that? Felipe, are you okay with where Lindsay found that? Good. How are polymers broken down to produce monomers? What kind of chemical reaction are you doing if you take something big and break it down into its pieces? Hydrolysis. Good job. We do count number 2. These are all parts of number 2. Number 3. What's a monosaccharide? Oh, we just wrote it. What is a monosaccharide?
Simple sugar.
Good. Okay, easy one. What's an example of a monosaccharide?
Glucose.
Ooh, that was easy. Glucose. I'll give you a page number in case you want one for reference, page 28. Define a disaccharide. 2 simple sugars. That's good. You tell me that. Kaylin, good job. 2 simple sugars. Your table's on fire over here. You girls are just good at this. You're ready for the test. What's an example of a disaccharide? Huh?
Is it maltose?
It is maltose. Everybody found maltose? Yeah, maltose. Again, what page is that? 28. Page 28. Okay, next one. Polysaccharide. What's a polysaccharide? Many. Good. Need an example? Good. Starch. This one's on page 29. If you want to look at starch, page 29. What is the common energy storage form of glucose in plants? Capital E means energy, and I want to start using it exclusively in our lecture on Thursday. Our lecture on Thursday is about energy in the cell, and so we're going to start using capital E to stand for energy as an abbreviation. So I went ahead and started today with that. What is the common energy storage form of glucose in plants? How do they store their glucose?
Isn't it starch?
It is starch. That's why we love eating plant parts full of starch, because you guys know starch is going to break down into sugar, which is good for energy. Sure, we love that. This question, what plant structure stored this substance, is so easy, I can't even believe I asked it. I mean, really, I can't even believe it. It's so easy, people don't know the answer because it's so easy. Does anybody know what we call plant structures that store starch? Yeah. We really did. Starch grains. Now, you and I were able to see these under the microscope one day. We did a really thin slice of potato, and we looked at it under the microscope. Do you remember this? We did onion, and then we did potato. Now, to see the starch, we had to add something to the potato, which I think is the next question. What must be added to potato to see starch? So easy. Can you believe Ms. Steinke asked you these things? Because it's so easy. Number 5's harder. This one's harder. Look at number 5. This is harder. What is the structural form of glucose in a polymer form in plant cells? So, look, they take glucose and they build it into a big polymer, but they use it for structure. So the answer is not starch, because that's for energy. I'll give you a hint. It starts with a C. It is cellulose. Great. What's your mama call cellulose? Your mama does not call it cellulose. What does she call it? She calls it fiber. Your mama calls this fiber. She didn't call it cellulose. Yeah, most people will call this fiber. Or if you look at a label for food, they're calling it fiber. It's the same thing. Should you be eating cellulose?
Yes.
Oh, yeah, you should be eating cellulose. Can you tell me 2 reasons why you should eat cellulose? Keep your bowel movements regular. Why else?
Energy. It digests.
No, it doesn't digest. That's the point. It makes you feel fuller, so it helps you with weight management. And here in the United States, that's a big thing. We were just talking about diabetes downstairs. Okay, so for weight management, cellulose is excellent. Okay, speaking of cellulose, I need a plant cell part that's composed of cellulose. A plant cell part made of cellulose. Anybody? Cell wall. Is that you, Denise? Excellent. Cell wall. It's structural. So eat more plant material. Lots of cellulose. It's good for you. Okay, number 6. What term describes the bond between two amino acids and a protein? Excellent. Good job. Peptide bond. In case you need a page number on that, peptide bond is on page 33. In case you want to look that up or look at the picture of the peptide bond, it's page 33, top of the page. Ready? Okay, on number 6. Okay, I'm going to give you 2 test tubes of lipid, and you have to be able to determine which has an emulsifier. So I'm going to kind of do a drawing here with you guys. And first, let me tell you what page this drawing is from, because my drawing is, you know, mediocre at best. Page 35 and page 36. I'll put that over here to the side. So what we did, everybody, is we did 2 test tubes. They're supposed to be the same diameter. And we put oil in both of them. So oil was at the top, right here, oil. And then we put water in one of them and bile in the other. And we kind of shook them a little bit and held them up to the light. So check it out, page 36. You may have written yourself some notes. Test tube 1, test tube 2, page 36. Yeah, so which one has an emulsifier? It is test tube 2, the one with the bile. But how would you know?
Because you're trying to balance it.
Yeah. So when you shake the test tubes and hold them up, you should be able to know by what you see. How would you know? What?
The clumps.
The clumps. Okay, so let's say you shook this first test tube, shake it, and then you stop and you look, and what are you going to see?
The first one,
You're going to see big bubbles, and then the big bubbles go to the top and make a layer. So it's pretty much going to go back to looking like it did before, right? But if you shake the second one, and then you look, what are you going to see?
a bunch of small bubbles.
A bunch of small bubbles, because you're emulsifying it. Okay, so make sure you go back and reread what you wrote about page 36, the 2 test tubes. And you can, yes, you can pick up the test tubes on the test and shake them and look. Give them a shake and look to help you decide. You can do that. You can pick up any of the test tubes and hold them to the light if you're trying to figure out color. You can pick up the brown paper on the test and hold it up to the light to figure out if it's translucent or not. You can do those things. Okay? Ready? Okay, on number 7. Super. Okay, adipose tissues on page 37. This is my very poor drawing of it. Of adipose tissue. There was a better one. It's really bad. Page 37, there's a pretty good photo of adipose tissue. But page 37 is a better actual photo. I just did a brief sketch of it. What it looks like, kind of looks like popped bubbles. Looks like bubbles. Okay, now what about functions of adipose? I'm thinking there's 3, and you probably wrote them down on page 37. Give me a function of adipose. Okay, very good. And you guys know that lipid is very high in energy. So I'm also going to put E in parentheses there. Capital E, what does capital E stand for? Energy. Do you remember how many calories per gram there is in lipid? 9. That's good. What about protein? 4. What about carb? 4. So who stores energy the best? Lipid.
Do we need to know that? Like how many?
Thought we already had to know that. Wasn't that on the test last week? Wasn't that on the test last week?
I'm saying for like this test.
You have to know it forever.
Oh.
I can't believe you asked that. You have to know this stuff forever. When I meet you when you're 55, I want to ask you, Diamond, who's got the most calories per gram? And you better say, lipid.
I mean like the number, like 9 lipids.
4 protein, 4 carb.
Yeah.
Yeah.
Oh man.
Yeah.
Okay. Thanks.
Can you believe her? I know. Wow. Okay, that was function number 1. Function number 2, go ahead.
Insulation.
Insulation. It keeps us warm. Keeps us warm. Insulation keeps us warm. Function number 3.
Protection.
Protection. It's kind of cushiony. So we put lipid or adipose around some of our more fragile organs, like behind your eye is a bunch of lipid, around your kidneys, a bunch of lipid. Adipose. Adipose. Okay, last part. Where can you find adipose in your body? Under your skin, primarily under your skin. That's where we put lots of it there, under our skin. Under your skin. There's other places, but that's the primary place. But okay, on adipose. And again, a good reference is page 37. For that whole question about adipose, page 37. Okay, cell structure. Let's move ahead. And would you open your lab manual to page 40 and 41 and lay it out flat so you can see page 40 and 41 at the same time? Page 40 and page 41. Number 1, how do you determine if a cell is pro or eukaryotic? What do you look for? The nucleus. So if there's no nucleus, what do you get? What do you have there? Is that pro or eu? If there's no nucleus, that's pro. And if you see a nucleus, then what is that? Eu. And the reason I'm mentioning page 40 is because I believe we have a sketch at the bottom of page 40 that one is pro and one is eu. So that's a nice little page to remember to go back and study so that under the microscope you should be able to tell if a cell is pro or eu. Now we're on this lab about the microscope and all the slides we looked at. I want to tell you that there will be no slides on the test of anything you didn't look at. So I'm not going to whip out some new stuff on you. On the other hand, every single slide we did look at is on the test. I used everything on the test. Okay, so there won't be anything new, but make sure you study every single slide that we looked at. Go back and review what did the oscillatoria look like, what did the human cheek cell look like. Everybody okay with that? Super. What characteristics do all cells have in common? I'm going to get you started. All cells have DNA. Thank you. Plasma membrane and ribosomes. That's excellent. Now we're going to find out that cells have other things, but so far you should already know these 3 things. That all cells have DNA, all cells have a plasma membrane, all cells have ribosomes. But you're like, no, I know already, Ms. Steinke, all cells have proteins because they're in all the pictures, proteins. Ms. Steinke, I know all cells have ATP because it's true. So you already have figured out some of the other things all cells have. But these are the 3 things I want you to know for Thursday. Okay, be able to identify parts of an animal cell. Well, page 44. Some of you have been rewarded greatly for doing page 44 with extra points. Now go back and study it. Page 44, parts of the animal cell. Page 44. Is it done? Did you do it?
Yes.
Be able to identify parts of the plant cell. Page 45. Go back and review that. You guys know some things plant cells have, animal cells don't. Some things animal cells, plant cells don't. Can you list 3 things a plant cell has that animal does not? Cell wall, let me write that down. Who told me vacuole? Very good, Adam. The central vacuole. What else? I need one more. Chloroplasts. Sir? Did you guys have something else? No? Okay, I'm going to go back to number 2. I want to find 2 things that animal cells have that plants don't. Lysosomes. That's good. Lysosomes. Something else. Oh. Centrioles. Are we back? Yeah. It thinks I went to sleep up here. Sorry. Lysosomes and centrioles. Okay, so let's make sure that it's clear in your notes what you just wrote down. You wrote down 2 things animal cells have that plants do not. Lysosomes and centrioles. And then you wrote down 3 things that plant cells have, animals do not. Cell wall, chloroplast, and the central vacuole. Okay, I want to remind you just one more time before we move forward. Make sure you review all of the sketches that you did, including the Elodia, page 46, the potato, page 46, the onion, page 46. Back on page 40, you did your cheek cells, and we did oscillatoria. Those are all the wet mounts that we did having to do with cell structure. So make sure you go back and review those slides and the labeling that you did on those slides, because they're all on the test. I'm not making this up. They really are all there. This test, good question. This test is 50 questions. 50 questions. It's a good question. Okay, cell function is what we just did last Thursday, so this might be real familiar to you. Cell function is still under Lab 4. And guys, diffusion, osmosis, and active transport, this is question number 1. I'm going to defer that to our lecture notes from today. You can just go back in your we just talked about it downstairs, so I'm not going to use your class time up here on that. Go back and review what is diffusion, what is osmosis, what is active transport, and take a look at that. Now, if you look at question number 1, under this is on the back of your review sheet. Which of those examples are passive? That means which of these costs no energy? And so out of those 3 things listed here they are, I'm underlining them. 2 of those don't cost anything for the cell to do them. Which ones are passive? They're for free. Diffusion and osmosis. Yeah, those are for free. And that's what I mean by passive. They're not going to cost the cell anything. The cell's just using concentration differences to get the job done. Okay, so obviously active transport is not passive. Is everybody okay with that one? Okay, good. And in which do the molecules move from low to high? Active transport moves low to high. Remember, the natural way to flow is high to low. That would be like going downstream in a river. But to go against that flow, low to high, you're going to have to use some energy. So that one's active transport. Active transport. Anybody need anything repeated on number 1? Okay, number 2. In which would diffusion be faster? A drop of dye in agar, which is basically jello, or a drop of dye in water? A drop of dye in water would be faster. Why?
Lower density.
Very good. Now, you guys remember that if you are a small molecule, you can diffuse really fast. Small diffuses faster than large. What about temperature? Which one would have higher diffusion? High temperature or low temperature? High temperature. That's good. All right, and then we also said the steepness of the gradient would affect the rate of diffusion. More steep, it would go faster. Okay, number 3 says review the experiment with the dialysis bag immersed in the beaker. Now, what would happen depending upon the substances placed inside the bag and the substance placed outside of the beaker? Okay, so that was on page 48 and page 49. Page 48 and 49. But I like this little picture. You'll notice this picture's different. But take a look at my picture. You can tell what I drew because I'm really getting good at this. I got a beaker, and I put water in the beaker, and then I made one of those little baggies, and I put molasses inside the baggie. Now, molasses is a real thick syrup, in case you hadn't heard of it. It's a real thick syrup. And I want you guys to think about water right now, because water is going to move. Which way will the water move? Into the bag or out of the bag? Into. Who told me that? That's good, Jeannie. It's going to move into the bag. Because the bag doesn't have very much water. And the beaker has a lot of water. And so water is going to move from high to low. So what's going to happen to the size of the bag? It's going to swell. So I'm going to put bag swells. It's going to get heavier and bigger. What kind of environment, think hard about this, using the words iso, hypo, hyper, what kind of environment did we put this bag in? Hypo. This one's hypo. The way I have it drawn right now, this is hypo. High water content out there, and the water's going to move into the little pretend cell. Cell's going to swell. The bag's going to swell. So take a look at it. Make sure you're okay with what I wrote up here. This should seem logical. Should be logical. Okay, check this one out. What will happen? Okay, Diamond thinks the water's going to move out of the bag. Do you agree? I do, because look at all the water is in the bag, and hardly any water is out of the bag. So what's going to happen to the bag? It's going to shrink. What kind of environment did we put this bag in? Hyper. This one's hyper. Good. Okay, so you should be able to look at a picture like that and tell me what happens. You should be able to look at that and tell me what will happen. Thinking about movement of water. What do you call the movement of water across a membrane? Starts with an O. Osmosis. So what we're doing here is osmosis. Okay, I like number 4. Go ahead and star that one. That's a good one too. Would you glance over that question and notice that the word amoeba is capitalized and italicized? What does that mean? It's not a genus. No, it is a genus. Yeah, that's a genus. Who told me that, Lindsay? Good job. That's a genus. Now, I am handwriting this. You could tell I hand wrote everything up here. So I couldn't do italics. So what did I do to show you it's a genus? I underlined it. Okay. I have considering the function of the contractile vacuole in amoeba, what environment do you predict it lives in? Well, let me tell you a little bit about amoeba. This is my expert drawing of an amoeba. If you don't think it's so great, I'll go ahead and show you a better picture of amoeba. We do have amoeba in our laboratory manual. If you want to take a look at that, let me show it to you real fast. This laboratory manual has it all. Just, finding it is what we'd have to do. So amoeba, still looking? Yes, page 189. There's an amoeba, page 189. So if you didn't like my picture. All right, so I did a few labelings on my simple picture, just some things I thought were significant. See my big circle right here that's got nothing in it? That's my contractile vacuole. There is something in there. It's called water. So some people call the contractile vacuole a water vacuole because it's just full of water. Okay, and then the outside black line right here, I have the abbreviation PM. What do you think that means? plasma membrane. PM means plasma membrane. Thus, no cell wall. Amoeba has no cell wall. Okay, and then this dark colored thing that's pretty big, I have the abbreviation N. What do you think that is? Nucleus. So would amoeba be pro or eukaryotic? Eukaryotic. Is everybody okay with that? All right, so let me tell you about the contractile vacuole. Everybody listen. Contractile vacuole collects water from the cytoplasm, and then it moves to the plasma membrane, and by exocytosis, it spits it out. And then the contractile vacuole collects water from the cytoplasm, and then it moves to the plasma membrane, and by exocytosis, it spits it out. And then the contractile vacuole collects water from the cytoplasm. Are you following with me? And then it moves to the plasma membrane, and it spits it out. So considering the function of the contractile vacuole, what environment do you predict it lives in? And I gave you the three choices. Go ahead. What do you think? Why is it constantly spitting out water? Because it's constantly what? Why is it constantly full? Where's the water coming from? Hypotonic. The water's constantly coming in. Water constantly is coming in. So yes, hypotonic. Now what would happen to the amoeba if it didn't get rid of all this water? It would burst. Why? Because it has no cell wall. But okay with that. Hypotonic. And if that contractile vacuole does not do its job, you are one dead amoeba. Be an amoeba explosion. Okay, number 5. My inspiration for this was page 53. I remember this last week. We cut potatoes. You remember that? Potato cutting. Put them in 2 test tubes. Look at my great drawing of a potato. It's really good. See my drawing? Huh? Okay. All right, so in this test tube, I have 0% salt, which of course means that it's 100% water. And here I have 10% salt, which means it's 90% water. And I put potatoes in both of these. And we actually did do this. And we let them set for about an hour, and then we pulled the potatoes out and we felt them. One of them felt really good and hard, rigid, and one of them felt all bendy, squishy, bendy. So I want to talk about which one's which. Okay, so which one would be really good and hard when you pull it out? The 0%, right here. This one would be, here's the word, turgid. He's really good and firm and hard. And what kind of environment did you put that potato in, iso, hypo, or hyper, to make it become so good and hard? Nope, not iso. Hypo. Why is it so turgid? Because it is swollen. Swollen. The cell's swelled. I know you figured it out. Since last week, you have figured out that 100% water is always hypotonic. Always. To any living cell, 100% water is always hypotonic. Okay, then we put a potato strip into 10% salt water. How'd that one feel? Limpy, yeah. And so we used the word flaccid last week. Just reminding you about that word flaccid, F-L-A-C-C-I-D, flaccid. What kind of environment would get you a flaccid potato strip? Hyper. Because the cell's shriveled. Which one of these? We'll just call this test tube 1. We'll call this test tube 2. Which test tube up on the screen right now do the cells have high turgor pressure? 2. What's turgor pressure? I'm sorry? It's the pressure of the cytoplasm against the cell wall. So this one, test tube number 2, has really good high turgor pressure. What do plants like to have, high or low turgor pressure? High. So it makes them stand up, gives them structure. They love high turgor pressure. But okay, on number 5, and remember page 53 is where we actually did the experiment. Okay, number 6, I'm going to do a little vocabulary here on page 51. And you're going to find these. You don't have to write them down. You could just say page 51. But crenation at the top of the page is even bolded for me at the top of page 51 is when red blood cells shrivel. Now looking at the pictures on page 51, help me pick which picture on page 51 would show crenation, A, B, or C? Crenation is when red blood cells shrivel. Is that A, B, or C? It's B. Okay, take a look at B. Here's a better question, more applicable to our test. What kind of environment would cause red blood cells to shrivel? Hypertonic. Very good. Can you see through a test tube with blood in it if the environment is hypertonic?
Yes.
Can you? Look down at the bottom and see.
No, you can't.
But check your data. You don't have to guess. Your data's right at the bottom. Find your hypertonic tube. Which one was it?
The middle one.
The middle one. Could you see through it? No. Because the cells were still there, so they were blocking the light. They were shriveled, but they're still there. Okay, check out the next one. Hemolysis, you can find the definition of hemolysis. It's bolded for you up at the top. But here's where red blood cells swell. And you know what's going to happen to them, don't you? When they swell, what happens? They burst. Good. We'll call it hemolysis, which means red cells split. Psh. Kind of environment would cause that? High who? Hypo. Can you see through a test tube when you put red blood cells into a hypotonic environment? Check it out. Look at the bottom. Which test tube was our hypotonic environment? The last one, number 3. Could you see through that test tube? I don't know. Could you? Yes or no? Yes, but check out your data you collected on the bottom of page 51. Print visibility, I think you wrote the word yes. Last Thursday you told me yes for number 3. Is that true? Okay, good. But okay, on crenation, not cremation, crenation, one letter makes a difference, and hemolysis. Okay, good. Turgor pressure. I found this in your lab manual if you don't want to write it down. Turgor pressure, top of page 52. It's the pressure of the cytoplasm on the cell wall. Would you ever talk about turgor pressure for animal cells? No. Why not? They don't have cell walls. So turgor pressure is a topic that you and I will talk about when we're doing plants. Talking about plants. But okay, on turgor pressure, you can find on the top of page 52. Okay, I made up 2 sentences. Everybody take a look at them. See if you can fill in the blanks. Everybody check it out. Because I just wrote them myself. Turgid cell. A turgid cell would be caused by what kind of solution? High. Hypo. That's good. Everybody agree with Deanna? Okay, look at the second sentence. A flaccid cell would be caused by a blank solution. Hyper. You're on fire. You're like, bring it on. I want this test. But okay with my sentences? Okay, still on page 52, guys. We did 2 sketches at the bottom of page 52 of red onion. Do you have them there? The bottom of page 52. Do you have 2 sketches there? Go back and review those sketches of your red onion cells. And make sure you can tell a red onion cell that's in a hypotonic environment. Notice how full it is. And a red onion cell in a hypertonic environment. Notice how shriveled it is. So check out your drawings, bottom page 52. Why would an animal cell but not a plant cell burst when you place it in a hypotonic solution?
No cell wall.
No cell wall. Very good. Who told me that? Is that Adam again? Good job. It's an easy question. Does anybody need anything restated for cell function, questions 1 through 8? Okay, real quickly I want to talk to you about pH in cells. Oh, man. Just a second, I'll be there. Okay, so on pH in cells, first of all, I don't know if you know that everything that we did on pH in cells was in our lab manual. So I'm just going to go over to it, show it to you, page 54 and page 55. Page 54 and 55, you know what I don't see here is the definition of an acid. So let me tell you, an acid is a substance that gives off hydrogen ions. So you guys may remember an example of an acid that we're going to use a lot in this class. We've already used it a couple times. Is hydrochloric acid. This is straight out of our lecture notes. You probably remember this from studying it last week. An acid that gives off hydrogen ions. It's a substance that gives off hydrogen ions. Good question, Haley. Okay, so if an acid gives off hydrogen ions, what is a base? Accepts them. That's good. Accepts hydrogen ions. And next week especially, we're going to use a pretty good little example of a base. It's called sodium hydroxide. You guys heard of it before, sodium hydroxide? It's the base we're going to use next week. Do some lab work next week using acids and bases. Now, everybody knows the pH scale. Mine's pretty pathetic, but they have a nice one on page 54. If I told you you had pH of 7, what would be a word that could describe the pH of 7? Neutral. And then if you go below 7, what is it? Acidic. And if you go above 7, basic. And some people might call that alkaline. Everybody's okay on the pH scale? Page 54 is a great reference for that. Okay, what's a buffer? It's a chemical that, tell me, does it break them down?
Weakens.
Let's use the word neutralize. Can we go with neutralizes? Do you like that word okay? It's a chemical that neutralizes. Now, they can neutralize acid or neutralize a base. So I'm just going to put neutralizes chemicals, like acids or bases. A buffer, you can add acid to it and it'll neutralize it. A buffer, you can add base to it and it's going to neutralize it. So buffers, you'll agree with me, help maintain pH homeostasis. Yes? What's homeostasis? Internal regulation. Very good. Okay. Now, take a look at my graphs up here. I like them. Miss Steinke, she likes graphs. Do you like graphs? That's too bad. I like them. And so take a look. I got graph A and graph B up here. One of these graphs is for a buffer. And this goes with question number 3. What is a buffer? And would you expect a curved or a straight line graph of pH values using a buffer? So this is the graphs that go with question number 3. Would you say A is a graph that shows a buffer or B is a graph that shows a buffer? Well, buffers tend to neutralize pH changes. We're putting in acids right here across the x-axis. Everybody see we're putting in acid across the x-axis? So which one of these is neutralizing the acid? A or B? B. So which one's the buffer? B. B is the buffer. You'll notice at the end the buffer becomes overwhelmed, doesn't it? But for a long time the buffer's working. Okay, A, it didn't do anything. You put in acid, it came down. You put in acid, it came down. You put in acid, it came down. The A is not buffered. It just came straight down. Okay, so B is my buffer. And if you need that graph, go ahead and draw that. That's fine. If you'd like to have that graph. This is the straight line graph. No, A, that's not a buffer. B is the curved graph. And yes, B is my buffer. A would be like water. Straight down. B would be like cytoplasm. Buffered. Question?
Oh, sorry.
Stretch it. Everybody okay on these graphs? Super. And then the last question is, what's the function of an antacid? What do antacids do in your stomach? When you eat Tums or Rolaids, what do they do?
Same thing, neutralize.
So basically they're buffers, right? Antacids are buffers. Okay, so let me tell you something.